Welcome to my lab. I'm Drew Collip. In today's lab, we're going to subculture BHK cells. Subculturing is a necessary technique in any tissue culture lab. You can see here we have a biosafety cabinet or vertical laminar flow hood. To turn it on, we raise the sash up to the prescribed height here. Then we turn on the blower. Fans will come on. Press the button firmly until it beeps and you see the symbol "Please wait." You now have to wait for the fans to reach the required speed. Let's get started. First, let's get some gloves on my hands. We go into the main lab. Gloves are located on this shelf. Make sure you grab the appropriate size glove. They're single use, so when you're done with them, they go in the garbage. Now we need a bucket. Anything that comes in contact with the cells or the media must be decontaminated. We'll use a mixture of a bit of soap and a little bit of bleach. Don't use too much. We'll then add about 2 inches of water. Please note, all pipettes should go into the solution. Never place a glass pipette into a plastic bag. The plastic bag is for non-contaminated waste and will be taken out by the custodial staff. If left in the bag, it could poke through the bag and seriously injure the custodial staff. Here we have sterile pasture pipettes. Notice they are cotton plugged to prevent aerosols. I'm also going to grab a 250 ml beaker. They should be located on this shelf. Don't get a smaller beaker. The opening will be too small and you could risk hitting the edges. If you get a larger one, you have to raise your hand up higher inside the hood, leading to muscle strain. We'll take these items back to our biosafety cabinet room and we'll check to see if the fans are up and running. We look at the screen. We see the "Please wait" symbol is off, so we can now sterilize. We'll place our items on the large stool and we'll sit on the short stool. Again, double check to make sure the "Please wait" symbol is off. It's also a good idea to hold your hand over the vents to see if it's sucking air in. We can now sterilize. We'll grab our paper towels and a bottle of 70% ethanol from the shelf. Everything that goes into your hood will be sprayed down with 70% ethanol, as opposed to isopropanol. For our non-contaminated paper and plastic wrap, we will use just a kitchen bag taped to the side. Again, no pipettes should ever go in this bag. If you are holding your pipette controller in your right hand because you're right-handed, then your bag and your bucket will go on your left side. If you are left-handed, the bag and the bucket will go on your right side. I'm going to pull off a large strip of tape here. And I'll use 3 pieces of tape to tape the bag to the side. I'll want to tape the bag as close as I can to the far side of the hood. I will use 2 pieces of tape to hold it to the hood in case it gets heavy with items being dropped inside. Be careful not to cover the vents at the front. The 3rd piece of tape, I will fold this over and tape it so that the bag stays open. Like this. Now I can drop items in without touching the bag. My bucket will be located just behind the bag. Notice, left versus right side. If you're left-handed, switch the sides. Again, double check. The "Please wait" symbol is off. Now I can sterilize my hood. I'm going to tear off a piece of paper towel and fold it into a pad. I will then take my 70% ethanol and spray the entire bottom plate with 70% ethanol. This will sterilize the hood. I will then wipe, moving back and forth from the back of the hood to the front of the hood, pulling contaminants out. Because this is paper, it's not contaminated, it can go into our bag waste. See how I can drop items in without touching the bag. Next up, all items must be sprayed down with ethanol and gently placed into the hood. Be careful not to spray these items over top of the common walkway. The ethanol on the ground can lead to a slip and fall. We'll grab pipettes now. We have 10 ml pipettes, serological, and 5 ml pipettes. I'm going to grab a handful of each. If I need more, I can go get them. If I take too many, I can bring them back. Please don't place too many in the hood. We spray them down and toss them in. Next up, we're going to grab a test tube rack. Very important, you spray the racks separately. Do not spray them with stuff inside. We spray the outside of the rack, and then we spray the inside of the rack as well. Again, don't spray it with tubes in the rack. Spray each separately, combine them together once they're in the hood. Pass your pipette bulb. I want to save my cells at the end, so I'm going to get a snap cap test tube. Notice it is sealed up with tape. It is sterile on the inside. Now we'll spray it on the outside with ethanol, and now it's sterile on the outside. Remember, never open these bags outside of a sterile biosafety cabinet. We'll now get our solutions. We're going to need our media, MEM. It's finalized, it's cherry red. We need PBS, phosphate buffered saline, and we need our trypsin enzyme to remove the cells from the bottom of the flask. Once again, we will spray these down. When you spray the bottles, be careful. Ethanol in glass is very slippery, so I will grab it like an eagle talon at the top of the cap. If you drop this bottle, you'll be back in the lab making more media or PBS. I think we have everything we need. Let's set up the hood and organize it. First things first, take off any watch bands you might have. Potential source of contamination. Bring your sleeves up. You have gloves on your hands. Now we spray our hands with our 70% ethanol. Now everything is sterile. We can go inside of our hood. Let's organize it now. I place my solutions on the left. I place my pipettes on the right. Make sure you don't block the back vents or the front vents. Have them off to the side. This is my waste beaker. I'm going to store it right in the middle. Notice how I open this. I will not stick my hand inside. I will shake one out to the edge and then grab it. I will then fold it over to maintain the sterility of the tubes in the bag. I will mix my solutions up by inverting them. I will then loosen the caps. I'll need to open these with one hand. If they're tight, you will not be able to open it with one hand. Now when I'm subculturing, I'm going to use PBS, then trypsin, then MEM. So I'm going to place them in that order. PBS first, then trypsin, then MEM, to help control the timing so I don't mix up my solutions. It's always a good idea to take things out of the hood you don't need. I don't need the bag anymore. I will fold it over, bring it outside the hood, and I will use this extra tape on the top to close it up and make sure it remains sterile. I will then place it on the shelf for someone else to use. If you contaminated your item, please don't place it back on the shelf. Here we have our pipette controller. Blow out, suck up. We look at the top, we have blow out, we have expel. You're going to set it to blow out. You can also adjust the speed of the pump with this bottom button here. I prefer 4 to suck up and 5 to blow out. If you're starting out, I recommend a lower speed. Notice the pipettes will come off this way with one hand and this way with the other hand. So depending if you're left or right, make sure you're using the proper technique. I'm right-handed, so I'll hold it in my right hand and take it off with my left. It must be sprayed down as well with ethanol. Make sure everything is organized the way you like before you get the cells. We have a test tube to save our extra cells at the end. Okay, now it's time to get the cells. These are BHK cells. They stand for baby hamster kidney cells. They've been growing for several days, and they're probably reached 100% confluency, meaning the entire flask has been covered with cells. We must now subculture. The protocol is written on the board here. You can't see the top part, my apologies. The first step is to check your cells. We will then remove the waste media and wash with PBS, then trypsin. Here's an inverted microscope. Notice the light on the front turns on here, and it is inverted, meaning the objective lenses are on the bottom and the light source is on the top. Set it to the lowest magnification. This can be adjusted, but always start on the lowest magnification. We have our light source. We also have a camera. We can switch this over between our ocular lenses and a television we have in the laboratory. The eyepieces, they're binocular and they're adjustable depending on the spacing of your eyes. Here we have our 37-degree incubator, and our BHK cells are growing in these flasks. These are T25s. Notice I hold them upright. Always carry them upright, not horizontal. I don't want to get the sterile filter at the top wet. We do a 4 C's check. We check the color, we check the clarity. It should look orange and it should look clear. Those are your first 2 C's, color and clarity. Your next 2 C's are cell health and confluency. The cells should look elongated and stuck to the flask. And what we should notice is every day there's more and more cells, meaning the confluency, the percentage of the flask that's covered with cells, should be going up. We focus in with our knobs on the side here. It always looks better when you look directly with your eyes in the microscope. But for this lab, to show you, I will output the image to the screen in the lab. Here we have our BHK cells. I've zoomed in. You can see they are elongated, they're stuck down, and they're touching each other. This is about 100% confluency, maybe 98%. We can zoom in a bit closer on the next objective lens, and you can see now, here are those cells. They look nice and healthy, very elongated, not rounded up at all. We can zoom in on our highest magnification, and we can look and we can actually see some of the nuclei here. So here's those BHK cells. Again, it looks better when you look at it directly in the microscope. So we're going to take these cells off the surface. We're going to take most of them out and only put a small subpopulation back. They will then continue the growth. Make sure you check all 3 flasks. You want them to all look the same. If one's 100%, one's 50%, and one's 10%, that's bad technique. I call this one consistency. So my 5 C's, color, clarity, cell health, confluency, and consistency. Make sure you turn the light off when you're not using the microscope. We'll now bring the flask back. Notice we don't carry it like this, we carry it upright, so we don't splash it on the cap. The only thing we don't spray are the flasks. I don't want to wet that sterile filter in the cap. If I do, air won't go into or out of the flask. We'll now spray our hands with ethanol and we'll get started. Now the BHK cells, they grow on the bottom of the flask, right here. I'm going to hold it at a 45-degree angle upright, so the cells will be up here. So if I put something inside, I won't scrape the cells off the bottom. I will loosen all my caps, and the first job is to remove the waste media. I will do this using a pasture pipette. Only grab the end there. Do not grab the part that's going to go into your flask. So this media, it's got a lot of waste in it from the cells, and it's lost a lot of its nutrients. In addition, any dead cells will be floating in there. So I'll remove this with the pasture pipette. I will place this into my waste beaker, making sure not to touch the side of the waste beaker. I will remove every drop I can of this media. The cells will remain stuck to the bottom of the flask. Now because it's all the same and I haven't contaminated my pipette, I can use the same pipette to remove the waste media from all 3 of the T25 flasks. Take your time, make sure you don't spill. Now when I'm done, I'm going to remove the pipette from the pipette bulb. If you do this inappropriately, you might get a splash coming off. So what I will do at the end is I will touch the pipette to the side of the beaker to get that last drop off. Touch it to the side, that last drop will come off, and I can remove it without contamination. Again, don't do it in the middle of the procedure, only do it at the end. Our next step is to wash with PBS. PBS will wash away any excess media. Make sure to loosen your caps first. I'm going to add 2 mils to each of these T25s. I have a 5 mil pipette. I will drop 6 mils and dispense 2 mils to each of the 3 T25s. Make sure don't contaminate the pipette. I unwrap it, I use it, I will get rid of it. Loosen the cap and hold the bottle at a 45-degree angle, like so. I will drop 6 mils. I will put my pipette over my beaker in case drips fall in, and then I will dispense 2 mils to each of these T25s. And it's only an approximation. If you overshoot or undershoot by a little bit, it won't make any difference. We're just washing away any excess media that might still be in there. The MEM contains calcium ions. Calcium ions can bind to trypsin and inhibit it. We don't want that to happen. Washing with PBS is just one way to prevent that. We will reuse the pipette and hold the overtop of the waste beaker, and then I'll take the flasks and I will make sure the PBS rinses all the cells. Using the same pipette, I will now remove all of the PBS and place it into my waste beaker. As long as I don't contaminate in between, I can use the same pipette for all 3 flasks. Once I'm done, I will then touch the side of the pipette to get off the last drop as to not cause contamination. I will take the pipette off, keeping the pipette controller inside the hood. See? Stayed inside the hood. I'm now done with PBS. I will seal it up. Our next step is doing a 2 mil wash with trypsin. We'll do exactly the same technique. 6 mils up, 2, 2, and 2. Notice how I open my Sealogical pipette. I push it through the paper side. Again, never put your caps down. Open it, keep the cap in hand, remove your liquid, and cap it immediately, holding your pipette over your waste beaker in case drops fall in, like that. We'll add 2 mils to each. Again, it's only an approximation. When we're done, we'll save that pipette and we'll rinse the cells as we did with PBS. I'll hold the pipette over the waste beaker and then rinse the cells. This way, we're not wasting resources. I can now use the same pipette to remove it. Now, unlike the PBS, I'm not going to remove 100% of the solution. I added 2 mils. I'm going to remove approximately 1.5 mils. Why? Well, the cells are going to come off the flask at this point, and I want some place for them to go. If I remove all the liquid, they may not come off effectively from the bottom of the flask. So I will leave behind a small amount of my trypsin. Why am I doing a trypsin wash? Well, some people just incubate in 2 mils of trypsin. I don't do that because I'm concerned about over-trypsinizing the cells. That's when you destroy all of your integrin proteins on the outside, and the cells will have a hard time binding to the new flask. As a result, I do a wash with trypsin. The trypsin is not active right now. It is an enzyme. We'll place it at 37 degrees for 2 minutes for it to work on the cells. There's a clock in the room with a second hand. And you can watch the second hand. It's at the 12, so I have to wait for 2 rotations around the clock. While I'm waiting, I can do other things inside the lab, such as removing items I don't need from the biosafety cabinet and restocking supplies I may have used too much of. Make sure I spray my hands when I place them into my hood. I'm done with PBS, And I'm done with trypsin. Let's remove those from the hood. And I'm done with my pasture pipettes. Anything that doesn't need to be in the hood is a potential source of contamination, so remove them. PBS and trypsin, I'm going to store them in my fridge, 4 degrees. And my pipettes, I'm going to reload them. There's a box here. Make sure there's 4 pipettes in that test tube, cap it, and place them in here. Make sure you're not using these pipettes for your work inside the hood. They are not sterile. I'm going to grab some more serological pipettes. Make sure I spray those down. And place them into the hood. Now what's happening now? The trypsin enzyme is working on those integrant proteins, holding the cells to the extracellular matrix. The trypsin, I don't actually want the trypsin to remove the cells from that extracellular matrix. I want them to be weakened. I want to look at the cells. I want to see that they're rounded up. I will then slap the side of the flask, and that force will cause the cells to come off the flask. Again, this is to help prevent over-trypsinization of those cells. Please wait the full 2 minutes. Now hopefully you remember what they looked like initially. They were coating the entire flask and they were elongated. You will now see the difference. 2 minutes exactly now. Let's take them out. I call the next step check, slap, check. Check them first. See that they're rounded up. If they're not, put them back in the incubator for a while. You will then slap the side and check that they've come off the bottom of the flask. See that? The cells have rounded up. They're not elongated like they were before. The trypsin has done its job. Check all 3 flasks. As you can see, the second flask, also the cells are rounded up. Now the third flask, this is the one that was sitting on the metal plate. It always tends to get a bit more heat, so it usually is less stuck to the flask itself. So trypsin is good. Now I'm going to slap them. I have big hands. I can do it all as one big grouping. 2 firm slaps. If you have smaller hands, you can do it one at a time. Notice I'm hitting the side, but also my hand. Don't hit the flask too hard. You'll crack the plastic. 2 firm slaps is all you need. So we checked it. We slapped it. Now we check it again. See the cells? They're no longer attached. They're floating in solution. I can pick up and drop the flask, and you can see the cells are moving around in that small amount of trypsin I left behind. Check all 3. Whole lot of cells there. And check the last one. You can see the cells are completely off now. At this point, I want to deactivate the trypsin. Again, I don't want to over-trypsinize the cells. So I'll bring them back to the biosafety cabinet. I'm going to add MEM to them. Remember, the MEM contains calcium ions. Calcium ions will now bind to trypsin and deactivate it. So please don't wait too long to deactivate your trypsin. Using a 5 ml pipette, I will suck up 6 ml and dispense 2 ml to each of my 3 T25 flasks. Open the cap, hold the bottle at a 45-degree angle, making sure I keep the cap in my hand. Take the solution and recap immediately. I will then dispense 2 ml of my MEM to each of those flasks. Again, I will save that pipette, and I will make sure the MEM deactivates all the trypsin by moving it around, rinsing the cells inside my flask. Now, most of the cells will have been detached from the bottom of the flask. Some might still be stuck down. So I'm going to draw up the liquid, and I'm going to spray it down. I'm not scraping the bottom of the flask with my pipette. I'm just spraying gently the media and the cells onto there to get any cells stuck in the corners off. That's extremely important. If you leave too many cells behind, you'll be seeding back way too many cells, and you'll end up having the subculture the next day. You want to be in control. I will then remove everything from that flask and pool it together. Now I have 4 ml in this flask. Again, I will rinse those cells off the bottom. I usually do it about 3 or 4 times. I will then suck up all of the cells and the media, and I will pool it all together in the final flask. As you draw the liquid up, make sure you don't suck it up into the pipette controller itself. Now all the cells and all the media are in this final flask. I will rinse the cells off the bottom, and then BHK cells have a tendency to clump together. So I will gently pipette up and down several times to try and break up those clumps. Now there may be some bubbles here, we may be losing some cells, but I think it's important we break up clumps of cells. I will then draw everything out. Every drop I can will come out of that T25 flask. Now is the subculture part. Everything's out, I will seed back 10%. I've added 2 mils in, I'm going to seed back 0.2 mils. How much you seed back depends on your confluency when you began. Because this was 100% confluent, I'm seeding back 10%. If it was less confluent, I would seed back perhaps more cells. When I'm done, I can either throw all those cells away, or I can save them to set up some experiments. I will save them in a snap cap test tube. Remember, if you throw them away, they're gone. So if you need to keep them, double check. Put them in a snap cap test tube, leave them until we're done subculturing, and we can do many different things with these cells. Again, touch the side of the beaker, remove your pipette, place it into your bucket. Now, we're going to put back MEM for the new cells to grow in. This 10 ml pipette can actually measure 14 mils. I'm going to load 4.6 mils into each. Ideally, you're loading back 5 mils, but to try and save on resources, I'm going to load back 4.6. I can use one pipette. So I will draw up 14 mils right to that orange line, making sure I don't contaminate that plug at the end. I'm going to load in 4.6 mils into each of these flasks. So I'm going from 14 down to 9.4, then down to 4.6. This way you've dispensed 4.6 into each, approximately. Touch it to the side, remove it, place it in your bucket of bleach. Seal the cap up so we don't spill it. And now we'll take our cells and put them back in the incubator. Notice the color of the media. It's not orange right now. It's this red pinkish color. As it interacts with the carbon dioxide inside the incubator, it will turn orange. Before we put them back in the incubator, we check them to make sure there's cells inside. Now, there's only 3 flasks here, so it'd be hard to miss one. But if you're setting up 50 or 100 flasks, it'd be easy to miss one. So I always double check all my flasks before I put them in the incubator. Notice those large circles, those are bubbles. The small white things, those are the cells. You can see there's cells in all. Now I'm going to put them in my incubator. The way I do this is called putting the cells to bed. I must distribute the cells evenly amongst the bottom of the flask. I place my caps to the back, and then I move them back and forth 4 by 4. Move them 1, 2, 3, 4 back and forth. 1, 2, 3, 4 left and right. 1, 2, 3, 4 back and forth. 1, 2, 3, 4. Stop. These cells are now evenly spaced in the bottom of the flask. Do not touch that for 12 hours while they adhere. Make sure to turn your light off. Now we'll clean up our hood. We have now finished subculturing. However, cleaning up is part of the job. Please put the pipettes back in the appropriate bins. 5s and 10s. They were not open, so they're still sterile on the inside. Pipette controller, give it a shake. If it rattles, it means the valve is open. You have not contaminated. If it doesn't rattle, make sure you're checking or cleaning your pipette controller. Do not leave it for someone else. This goes in my pocket. And this MEM goes in the fridge. 4 degrees. My waste comes out and I pour it into my bucket of bleach. Turns purple, and then it disappears. That's that phenol red inside the media. Here's the cells. I'm going to use them for another experiment. And I sterilize the hood at the end the same way I did at the beginning. I'm going to tear off some paper towel, fold it into a pad, spray the entire surface with 70% ethanol, and wiping back and forth, moving from the back of the hood to the front of the hood, pulling contaminants out of the hood. Make sure you have a nice mist on your spray bottle. I want to have the hood cleaner when I'm done than when I started. There's nothing in here but paper and plastic that's not contaminated. There are no pipettes. No glass to stab me. Close the sash. It will turn off the light and the blower. Push in your stools. Put back our ethanol and our paper towels. And we'll dispose of our items in the main lab. This can go in regular garbage. Remember, never any plastic or glass pipettes in there. Over here we have some biohazard bins. We'll place my plastic pipettes in the bin. Glass pipettes go in this one. I will then rinse out my beaker. It's been sitting in soap and bleach, so it should be pretty clean. Let's try and get some of that off. I'll then rinse my bucket out. Please try and rinse out the bucket. Don't leave the bleach inside. Over time, eventually, the bleach, I think, will work at the plastic over many years. So let's rinse it out. Keep them as long as we can. And subculturing is now complete. I have these cells left over. I'm going to use them for my next video. I'm going to count the cells using different methods. This is one method here. This is called the culture counter. In my next video, I'll show you how to do that. That's all for today. We'll see you in the lab.